Good afternoon. Thank you for joining us for this webinar to introduce Stereo Investigator AI. My name is Alyssa Wilson, and I am one of the product managers.
And I'm Jorge Castro. I'm the other product manager of Stereo Investigator AI. As Alyssa mentioned, we are really pleased to introduce to the audience Stereo Investigator AI. So during this webinar, we will explain what is a Stereo Investigator AI, advantages of using this software over classic stereology methods, and how the software works in a brief presentation, and then move to an actual demo showing the software, how to set an experiment, validate results, etc. Finally, we will move to a Q&A segment where the audience can make questions. And actually, if you want during the presentation, we'll have an open chat. So people can post questions and we will be answering those at the end. Okay, so to define a Stereo Investigator AI in one sentence, we can say that it is a program that combines advantages of using design-based stereology with deep learning AI to count cells in a sample. But just to give like a step back for those in the audience who are not familiar with stereology, let's very briefly introduce the concept of this technique called stereology. So stereology is a technique based on statistics and geometry that allows like very efficient and unbiased estimations of cell number, volumes, and surfaces. Now, it is considered efficient because it does not require to sample your entire region of a study. You can sample a fraction of your sections or the area and yet produce an estimation of for example, number of cells with a coefficient of error that provides the researcher with an idea of the accuracy of the estimation. And it is unbiased because due to the way it samples, the particles or objects you want to count will have equal probability for selection. So there is no bias by size, distribution, or form. That is what this graph is showing here, that independently of the direction of sampling, the number of counted cells is going to be exactly the same, even though the distance between optical planes or the number of optical planes are different. So with the Stereo Investigator AI, we are taking the stereology sampling techniques. To be more precise, we are using the Optical Fractionator probe. But so we don't, the idea here is that we don't need to analyze the entire sample, but we still provide to our automatic cell detection tool an unbiased selection of the cells we want to count. Now, stereology has been so useful that has been adopted by, like neuroscientists and researchers in other fields, like, like anatomy pathologies, for many years for quantification purposes. And it is considered the gold standard for quantifying metrics, like number of cells, volume of organs, like brain regions, etc. That's why to help the researcher, the research community, NBF developed a program to use stereological principles in their study. This software is a Stereo Investigator, and it has been in the market for quite some time. It's currently used by hundreds of labs and has been cited by more than, like 8,000 publications. Now, despite being a very efficient technique, so again, like no need to count all your regions of interest, with a Stereo Investigator, there is a human annotator or counter doing the, the counting. As we can see in this video, a person will need to navigate through the sample and decide what is considered positive versus negative. We'll just go through like, like all this stack, etc. Like, like make the call if, they should just place a marker for these. So this makes this technique very labor intensive when compared to current automatic cell detection methods, and also prone to human bias. In fact, researchers working on the same project need to make sure that they are counting their population with the same criteria of selection. Because this requires training and a certain learning curve. Once a trained researcher moves to another lab, a new trainee needs to be trained, and again, make sure that, like criteria used for counting are kept consistent. So there is this problem of liveness in the lab expertise. So to overcome the shortcomings of a human-operated Stereo Investigator, we decided that we could apply the new developments in deep learning AI to create an automatic cell counter. that will still be bound by a stereological principle. So what are the benefits of Stereo Investigator? When compared to classic manual stereology methods, it obviously reduces the labor to obtain cell counts and also increases the throughput. Actually, in this image here, we are showing the project definition window in Stereo Investigator AI. You can see how a user can easily set a detection queue or project run to count cells in multiple sections, multiple regions, and sites represented by these checkboxes. This process is extremely simple to create and will be explained later in the demo part. Additionally, the process is perfectly repeatable. So multiple detection parameters can be tested on the same locations to see what combination can produce, better detection results. And as I mentioned before, the sampling is unbiased. Now, what are the benefits of Stereo Investigator AI when compared to exhaustive automatic counting methods? We know this is not the first software that does automatic cell counting. In fact, in this video here that was created with other MBF program that has the capacity to do automatic cell detection in an entire region based on pixel intensities. The software is called Neuralink 360. So the problem of trying to do a cell detection in a massive data set, for example, trying to do cell count in an entire brain, is that after you do the automatic detection, you want to audit and validate those results. And with a huge data file with hundreds of thousands of cells, validating these results in a structured ways is really impossible. And at the end, and I'm speaking for myself when I'm using Neuralink 360 to do some counting for customers. You see how the detection has worked in some locations and take the results produced at face value. But with Stereo Investigator AI, because we are already sampling a representative but unbiased population, we can properly audit and validate our results and being confident that if the detection is done correctly, we can trust our result. Actually, we will see later that we have a built-in validation workflow to check how precise the detection has been. Additionally, because we are applying the detection in a subsample and not the entire region of interest, there might also be some, speed performance advantages compared to exhaustive automatic counting methods. We also have to take in consideration that these exhaustive automatic counting programs can have precision problems or show, false negatives. As you might have seen in the previous video, there were a few cells that were clearly positive that they were not marked, so they were false negative. But as we show in a presentation this year at the Society for Neuroscience, our detection performance was very good, especially discriminating false negative. In terms of sample preparation and imaging requirements, the Stereo Investigator AI is not very different from the Stereo Investigator. We do quantification on previously acquired fluorescent images, and hopefully soon we will also add bright field detection too. Sample thickness requirements are exactly the same as with the Optical Fractionator, which is the specific stereology probe that we are using with this software. So you need samples that are usually thicker than 15, 20 microns. We don't need very high lateral resolution. In fact, beta testers data was primarily acquired with mid-magnification objectives, 20x, and step sizes of 0.2 to 1 micron. And in terms of imaging modalities, we've been testing wide field and confocal images, but although we haven't really tested yet, I don't see why it should not work with other imaging modalities, like shade to photon, other than the need for a relatively short distance between optical planes. So, how does the software detect cells? Stereo Investigator AI utilize deep learning engine that is based in custom convolutional neural network architecture, CNN for short. that utilize GPU-based acceleration. The system, as we'll see in a moment, can operate on partial intensity projections in the XY, XZ, and the YZ planes, or in individual focal planes, and it will combine the output into like a 3D volume. The CNN can accurately locate multiple objects of various sizes within a field of view using 3 parameters or elements. The 3 elements that the deep learning engine will use are listed here. Okay, these 3 elements can be modified by the user, also something that we will see during the actual demo of the program. These elements are first pre-trained classifiers that we have developed in-house using data from research collaborators. And second, it's different detection methods that provide the classifiers with candidates for classification. And third is a size range selection that the software will use to eliminate candidates that are like well outside of the size range. So the first element are the pre-trained classifiers. We have a wide variety of classifiers from, like generic, like basic round shape objects with no very distinct features to very specific for a particular stain or cell type. For example, distinctive somatic only stain like in TH, or more punctuated and with a particular shape like like microglia here with IBO1. In any case, we are very open and eager to add more classifiers. So if you don't see a cell type you would like to quantify, let us know and we'll try to create one for you. Right now, all new classifiers are trained in-house, but with end user feedback, of course. The reason we are training the classifiers in-house is that at this point, it's easier to provide better results by getting feedback from the end user and incorporating that into the classifier by one of our expert developers than really the end user dealing with refining the classifier by themselves. The second element in the Stereo Investigator AI to do the cell counting are these different detection methods or ways to generate 3D objects for presentation to the classifier. The first method is called the LOG or Laplacian of Gaussian seeded detection method. It uses a multi-view fusion algorithm that combines slices along the X, the Y, and the Z axis, creating like a simple free plane presentation like here. This method is very efficient and fast because we are leveraging the LOG detector output and restricting the detection on specific ROIs. But unfortunately, it works best with like high axial resolution imaging methods. So it's only suited with confocal stacks. The second method is the XY scan, which creates 3D objects by using clustering techniques of individual detected XY focal planes. It is more flexible than the LOG seeded because it's not so sensible to out-of-focus illumination. So it can be used for confocal and wide field imaging, but it's slower and it's also less accurate to determine the Z position of centroids. And well, I mentioned centroids. Now that's something that I haven't mentioned before, but Stereo Investigator AI is using centroids as the counting particle. For the people not familiar with the stereology, one rule while counting cells is that you have to determine a counting particle that is unique, but also present in all the cells you're counting. The convention here in classic stereology is to use the top of the cell as counting particle, as it is like a very well-defined event when it starts showing up as you progress down the sample. Like remember the video that we had before where the human counter was like going up and down in the stack. So yeah, the person would just see like, when appearance of the top of the cell would happen and then would place a marker for that. So instead of the tops, Stereo Investigator uses the centroids. This really does not have any impact other than when we do the validation of what the software has counted, we'll need to specify if we are counting tops or as the computer is doing centroids. The final Stereo Investigator AI, the final element that the Stereo Investigator AI is using, it's a user-defined size range. We have called the parameter presets, and you can easily create one by looking through, like multiple locations and using a tool, which is basically a cursor that you can change the size using the mouse wheel to measure the longest diameter of the largest cell and the shortest diameter of the smallest cell. Finally, once the run or count is done, the software will provide with the same kind of report you would receive in the Stereo Investigator with, like your cell number estimation, different types of coefficient of error, etc. Obviously, during the first runs, to make sure the software is counting the right cells, you want to validate the results. And this is also something that we added as a workflow here. So the user can determine how many, like sampling sites wants to check, like kind of like, as a percentage. And the software will randomize not only the selection of the sites that you are going to validate, but also the order of the sites presentation, and will hide markers placed by the computer. So it will minimize, any kind of like user bias. After the user finishes, the software will provide a report with precision and recall values, which are telling you, like how accurately the software is detecting the cells. There is also a 3D view, like after you finish, like you have like this 3D view window here, which is very helpful to determine like edge cases like this one, for example. So here we see like, like 3 cells that the software has detected. These are the centroids, like these like tiny green dots. Then the mesh, the green mesh that you see here, that's kind of like a representation of like cell volume more or less. And then the yellow open circles, those are the markers that the software has placed. And in this particular case, like, like the software, like, was detecting the centroid, like really at the border of the sector. And, like you can always like use this view to like rotate, and especially for like, like, for example, determining like the Z, like if a marker is like properly placed, depending on the Z location, this view can be really, really helpful. And something that we didn't have in a Stereo Investigator. Okay, so to recap or summarize, Stereo Investigator AI is the first design-based stereology software to provide automatic true 3D stereological cell number estimations. It combines a Stereo Investigator widely used optical fractionator workflow with an easy interface to select different parameters for a successful automatic cell detection, and can create long tasks of, or projects encompassing multiple sections and regions of interest. The user can refine the detection output by changing parameters such as, cell size or the detection methods, accommodating different image modalities and different cell types. And in summary, with the Stereo Investigator, you will have a tool that can quantify cells with minimal effort, but it's accurate, unbiased, and very easy to validate. We would also like to mention that, to people that people are more than welcome to do a free evaluation of the software. We can discuss that later. And we would also like to thank beta testers for the data and, like very helpful input. And with this, I will hand over the presentation to Alice.
Thank you so much, Jorge, for going over all of the details. And now we will get to see it in action. And I will share my screen. When you begin, you will start out on the main ribbon, and under Get Started, you will have 3 icons to use. Whole region will be selected if you did a complete scan of your region of interest. This option will bring you through a workflow to set up your stereological parameters. SRS images is the option to use if you use systematic random sampling to collect your image stacks. An open project is the option to use if you are opening an existing project file that you would like to continue working on. Briefly, we will go back to the slide at the beginning of the presentation to go over some of those needed stereological parameters. Before you begin the acquires, you will need to determine the section interval. This will be used to determine the serial section fraction. You do not need to collect image data from every single section. So here the lines show all the sections, and then you're able to just image certain sections. Using the total number of sections within your region of interest, you can determine the proper section interval to identify the evenly spaced sections to include in the image acquires, as you are able to see in this graphic. For example, you may want to do every fifth or sixth section. The next parameter to be determined is the grid spacing. The grid spacing and counting frame will be used to determine the area section fraction. As you can see in this image, the highlighted grids indicate the sampling stack acquire location, or the systematic random sampling, determined by the grid spacing. If you are using a whole region acquirer, then you would be acquiring all the images shown in blue. So therefore you would be acquiring all of these adjacent images and stitching them together. This can be done using Stereo Investigator and Neurolucida and using the slide scan workflow. Or if you have a whole slide scanner that can acquire in 3D, that can be used as well. And lots of other options. We are able to support lots of different image formats. And in this case, the grid spacing and counting frame would be part of the setup for that whole region workflow and apply to this whole region. And therefore, the counting would still be completed on a systematically and randomly sampled image stacks. By doing the whole region, it does give you the flexibility to set up multiple SRS acquires on one image set. On the other hand, if you are using the SRS acquire option, the grid spacing would need to be done during the setup of the acquire process. For example, this can be done in Stereo Investigator using the SRS stack acquire workflow. And because you are only acquiring the stacks at each SRS site, so the ones that are highlighted, this type of acquire does save resources and acquire time. The dissector height is the final parameter to be determined. The dissector height will be set depending on how thick the tissue is mounted on the slide and will set the thickness of the stacks to be acquired. The dissector height and the section thickness will be used to determine the height sampling fraction, and the section thickness will be automatically measured at each site, so it is important to acquire images above and below the top and bottom of the stack. Now back to the demonstration. For this demonstration, we do have SRS images, so we will select that option. And there are two ways to move through this workflow. If you have SRS images that were acquired from an outside source would be any system other than Stereo Investigator. And you would also have to have acquired your image stacks systematically and randomly and with a known grid spacing. So selecting this option will bring you to a workflow that will apply the grid spacing and those other stereological parameters, and then take you through that workflow. The second option is if you have used the SRS stack acquire workflow in Stereo Investigator, which is what we have done. So that is the one I'm going to select. And when we browse, here it is prompting me to select an XML file. This XML file contains the sections with the contours around our region of interest and the associated SRS image stacks. So now we are loading in those images, and I'm just going to adjust this so we can see our whole contour here. So from this acquire, we already know the stereological parameters because they were set up during that SRS acquire workflow. So we just have a quick two-step workflow to go through here to set up the counting parameters. Here you will see the contour that was traced within Stereo Investigator to outline the region of interest in this one section. For this study, we are imaging nu-N in the motor cortex. The SRS image stacks are acquired within this contour of each section. All of the parameters are parameters that were set up during the SRS Stack Acquire workflow, which include the number of sections, our cut thickness, and our section interval that we talked about before. And there is also a note that the centroids will be used to determine the cell location, as Jorge had mentioned. And now we can move to the next step. So now we want to create a probe run and sites for counting. If you do have multiple sections, you can select that here if you want to do all of them, or if you just want to do one section. For this, we'll do one section. And if you also have multiple regions of interest, you can select which region you're going to do. For this, we will select our motor cortex. And then we want to select which channel we want to count. So in this case, our new N was acquired in the green channel, which is channel 1, and the preset needed. So this is the cell size parameter that we went over earlier. And here we have determined that our cells are 8 to 16 microns, so we will use that preset. And for the classifier, we are counting new N, and this is wide field. So we will use the new N wide field classifier. And as you can see, we also have the list of other classifiers that Jorge had mentioned earlier. And this list does keep growing, and we want to continue to have it grow. So looking forward to seeing your image data. And our detection option. So here again, something that Jorge went over earlier. And for this one, because we have wide field, we will use the XY scan option. And then we want to select a marker that will be nice and easy to see. So I will use this filled circle. You can also rename the marker if you would like to. And this chart here, it lists all of the stereological parameters. Again, these were set up during our acquire, but we have our counting frame, our grid size, our cut thickness, and our dissector height. So now we will add to the run. And now over in our project definition window, we show the probe run that we just created and all of the sites. So as we can see, we have section 5, which we selected, and the contour we selected. And then here is a list of all of the sites that are going to be visited in this section. And these sites are systematically and randomly sampled based on that grid spacing. So here, we can select the section that we would like to count. If you have added all of your sections for your entire study, you would just select the top box and it would select everything, and you would be able to count everything in one case. And at this point, you can just click count and walk away, which is great. It does prompt to make sure that you want to save all of your changes, and then it will initialize your image and get started. So at the top of the project definition, it does show you the status. So if you want to check in and make sure everything is looking good, you can see the status as it is going. So this section will take about 5 minutes. So for this demonstration, I will switch over to one that has already completed to be able to view our results. So now this is what it will look like when it is done. You will see in the project definition, the status is completed, and it will give you the total cells, the total cells and the total counted cells. And then we can also view our results. So if you have Stereo Investigator, this button will probably look very familiar to you. So our probe run list, so we can select that, and this gives us a list of all the probe runs that were created, and we can view the results. And here we can view our results and also export those results to Excel. And again, this is going to be the same thing that you get if you export your results from Stereo Investigator. You have your markers, the total number counted, your estimates, and your CE values. So going back, so now we have viewed our results, and now we can view or visualize our results and the markers that were placed. So in order to do that, we will select the section we want to view, go to our view ribbon, make sure we'll see our counting frame and markers, and then we can select this little eyeball here. And this will turn on the image at that site. Okay, so now we can see our image. We can see we have the 4 markers here, which is the counted cells, these yellow filled circles. And I am in the 3D view. So here I'm able to rotate, and from the side, we can see that it did a really good job of placing these markers in the center of these cells. Now, another thing we can view in the 3D window is the centroids. And now this shows us our total cells detected, which is the total cells here. But as you can see, only the cells that were within our counting frame are the ones that got the markers. So another view option that we have is 2D view. And we'll switch over to that. And here we can use our page up and down or our scroll wheel on our mouse to move up and down through the stack. So now that we have viewed and visualized our results, we may want to validate the results. If you and you can do this if you want to double check and make sure the counts done by the program match up to what you as a manual counter would do. This is an optional workflow that will allow you to manually count some of the sites. And while you are counting, you will not be able to see the markers placed by the software. So in order to go there, we will go back to our main ribbon and go into our validation workflow. So in step 1, we will set up our information. I'm just going to call this demo. And here we are able to select the number of sites that we want to audit as a percent. So here I'm going to put in 25%. We don't have that many sites, and update that. So yeah, so we'll just be visiting 2 sites, but obviously if you're validating a whole study, you can determine the percent that you would want to validate depending on that total number of sites. And the other thing is the program will randomly select sites for validation. So the program does that automatically. So we can go to our next step. And now we can select if we want to measure our thickness while during the validation, or if we just want to use an average thickness. So here I am going to select the marker that I will be placing. I'm going to use the star, and then the location of the markers. So am I going to be placing the markers at the top, center, or bottom of the cells? I'm going to use the top because that is what I am familiar with as a counter. And then go to our next step to start the validation. So again, this view is very familiar for those of you with Stereo Investigator. This is what it would look like if you were counting off of stacks. So we're going to go to the top of our stack. Oh, let's go to the top. And we're able to set the top. So we will set the top and then set the bottom of our stack. Okay. And now we are able to place markers on any cells within the counting frame. And once you finish placing your markers, you can move to your next site. And I will skip over this for now. But once you are finished, you can export in Excel showing the validation results. So this will show you the number of markers that you placed as the human counter. and that the program has placed. And then from there, you will also get your true positive, false negative, false positive with your recall and precision. These values are determined by colocalizing the markers placed by the validation and the software. So it does this by using the preset settings of the cell size. So knowing the software is placing a centroid and the validator was placing their markers at the top, it is able to determine which markers should be colocalized and were placed on the same cell to get that true positive value, or which markers were placed on different cells. So this would result in a false positive when the software placed a marker on an object that the person didn't consider a cell, and also the false negative when the software missed a cell that the validator did place a marker on. And so this is the end of our demonstration, and we can begin the question and answer portion of the webinar.
So we are receiving some questions here. There's one question about how to make a contour.
For the contours, that is something that would actually be done on your acquire system. So whether you're utilizing an MBF piece of software, which would be Stereo Investigator, or different software, your contours would be done there. Your acquires would also be done there. And then that data would be brought into Stereo Investigator AI to then set up the probe runs and do the runs. So that is something that actually is not done within this software, because all of the acquires are being done in a different piece of software. And that is definitely something that we can go over if you're interested in a one-on-one demo and how to do those contours and acquires on whatever system that you currently have. We could go into depth on for your individual needs.
Okay, perfect. We have several questions about bright field. We got questions about when bright field might be available. Also about development for missile stain section. So yes, we actually, as we speak, we are working on bright field image cell detection. As we are seeing in the in the chat box, yeah, there's a lot of interest in that. There are definitely some changes necessary to be able to detect cells in bright field, primarily the fact that it's a composite of 3 channels, and Stereo Investigator right now it's only using one channel for detection. So there is more information, and additionally probably the mix of these 3 channels that actually provides color information. It's already giving more features for this classifier to analyze. So most likely we'll need classifiers for bright field and the fluorescent version of the same cell type. So maybe for people interested in NICEL for NICEL for bright field, and then maybe neurotrace for fluorescence. And we cannot give a estimate, but we are really working right now on that. So probably in months we'll have something. But the one important thing is if you are interested, people asking about providing data for for example NISO, if you're interested, please let us know. You have this information to contact us. Please let us know. Send those images, and we can do some training on those images.
Yeah, definitely. I think that would be great to contact us. We'll have some after this, there'll be a form that you can fill out. So you can put in your contact information, and we can follow up with you afterwards and be in touch with, like Jorge said, different training data, and also yeah, how we're progressing on all that bright field work.
There are a few questions about how long.
Yeah. So we have found that your computer is a big, an important factor in how long it takes. On our website, we do have some recommended specs for the computer. But for example, the section that I had that had about 10 sites, it takes on some of the faster computers that we have, it takes anywhere from 5 to 9 minutes to run. So with some of the, less processing power computers, it would take longer. But the nice part is this is something that can run overnight. So, while you are home enjoying your personal time, the computer can still be working.
Yeah, to add on that, like the in terms of like specs for your computer, is going to be very heavy on the CPU and the GPU, not so much for RAM. Because we are leveraging like like GPU cores, like definitely you need to have like probably like one of the latest versions of like NVIDIA graphic cards. So but yeah, we can like we can discuss that on a one-on-one basis. If you want to try the software, like like we can discuss like what would be like a good build for for running. There is one question about the Stereo Investigator AI being compatible with live sheet images. So we don't think that like we haven't really like tried using live sheet images, but we don't think there shouldn't be like any problem other than like the distance between optical planes. So all the classifiers that we've been like testing, they were like in between like 0.5 to 1 micron. So yeah, definitely, like you need to have probably like a good axle resolution for, with your live images. But yeah, bring your images and we can, like we can try it out. Can one download the new program from the usual space? Right now, it's not in the download center, but yeah, we can like we can discuss later. Like if you are interested in a trial, we can provide you with like the software and the classifiers that might be like like in separate files, and yeah, like but right now that they are not in the download center.
But yeah, we are doing evaluations with image data, and if you have images that work with our current classifiers, you can get a trial. And if not, we will work with you to create classifiers. So definitely interested to see what images that you have.
We have another question about queuing up like like multiple slides and just leaving the software doing like the work. Yeah, absolutely. Yeah, that's the project run definition, one of the windows that Elisa was showing. Like you can just start like adding all the like sections, all the regions that you want to like analyze, just put that in a queue and then you just let the software run. So yeah, that's not an issue. And the kind of like the beauty here is like if you want to change parameters, so like you want to change like the detection method, for example, you click on one of the top boxes. Just think about these like kind of like a hierarchical, like you have like your project and then you have your sections and then you have like your regions. So if you want to change like all the detection method for all your sections and all your regions, you just click on the kind of like the project box, and then if you change the detection method on one of those, it will just like propagate through like all the project. So it's like super simple to use and very user friendly.
And there is a question about new classifiers that we're working on or classifiers that we plan to add. Definitely our big focus, like Jorge mentioned, is bright field. And then outside of that, it's, what is requested. So like I said before, we'd love to see your images, hear from you what you would like to count. So we want to work with you to develop those new classifiers. And then there's also wondering, besides cell, can the software count axonal boutons? So if it's something that we can train, then it is something that we could count. So definitely please get in touch with us and we can see if we can develop a classifier for that.
There's one
Oh,
about
okay. So stereo software. Um, So yes, currently SIAI is a standalone software. But please send us if you already have Stereo Investigator, please contact us because we definitely want to work with you to be able to integrate Stereo AI into your workflow.
There is a question about like doing the detection on two different populations, like new N, for example, and IB1. So yeah, we can handle these like like multiple marker, multiple population. You will need to do it like in multiple runs, like like one for like one of the classifiers for like let's say new N, and then the other one for the IB1. But you can just like kind of like put it in a queue and do like like kind of like first one and then another. So that's perfectly doable. And we have a question about like IVA1 and different stages. So yeah, we know about these. We don't have right now any specific, classifier for like different stages of activation. We'll need to like, like train that. Like probably there will be like, like different classifiers. But yes, we've been approached by some people about microglia activation state. So if you are interested in that, like really happy, like if you provide like the more data that you provide, the like the more robust the classifier will be with like extra training. So yeah, yes, please upload those like different IVA1 states. So thank you. So we are not gonna. So there is like questions about cost. We are not gonna, like you can just ask like contact us like like separately, and we'll send like a quote. We have people from like like different parts of the world, so just like saying numbers here like it wouldn't make sense for some other people. So yeah, yes, please contact us like directly to like like info at MBF Bioscience or directly to us, and we can provide like quotes for that.
And there's a question about the computer requirements. We have the recommended on our website. So if you go to the MBF website, we have a Stereo Investigator AI page, and the computer recommended computer specs are on there. So or you can please send us an email and we will be happy to provide that for you.
There's one question about the type of image files. Should I bring this 3D image files? Stack image files is not available. No, we can either, like if you provide us with like your whole region of interest in a Z stack, like kind of like a 3D virtual section of your region of interest, that's fine. Then we will just, like subsample. But you can also bring, like if you already have, like, for example, some of the Steam Investigator system, and you are collecting your images following SRS, and you have your XML file, you can just provide those stacks with the XML file, and that would be, that would be also useful for us. So any kind of like, like image modality, or in image type, it's fine. Like, either like, the whole region of interest, or just, like subsample of those. There's one question about other, like how we compare to other platforms like Aphoria. So I don't know much about these. Like I, like recently heard about this platform. As far as I know, like kind of like the main difference that I'm seeing is that the way that we train. So we are doing like the training in house, whereas in these cases, like you have to like, like kind of like classify, like the end user is like classifying the object. So that would be like kind of like one difference, I believe. But yeah, I cannot say much about like, about like, yeah, like how we just differentiate or like compare exactly to Aphoria.
And then there was another question about the demonstration and providing the software. So again, that will depend on your images. So If you reach out to us, if you're interested, we will do the evaluation on the images with our current classifiers, and then depending on the results of that, we can either do a trial, like allow you to trial the software, or um, work with you to develop a classifier that would work for your cell population.
And there's one additional question about quantifying expression levels using immunohistochemistry or RNA scope. So these, the technique that we are showing here is the optical fractionator is going to give you like an estimation of number of cells. We are counting particles. We we are not doing like kind of like intensity analysis to like quantify expression levels, if that's what you mean. So I mean, if particles could be kind of like a proxy for expression, then maybe. Obviously, the size of the particle, like that might influence the detection. What we've been like, training the software for is like kind of like, like cells. So, think about like, from like 4 to like 30 microns in size. But yeah, just to like make sure that people understand this, like we with this software you're getting estimation of cells, not level of expression like intensity, like what you're getting from like some colorimetric analysis or like RNA scope analysis.
Okay, I think that is all the questions. Like I said, please follow up with us. Like I said, we'd love to see your image data. Oh, another
There's
question
one.
came in. Yes, so right now the optical fractionator is the only probe right now, and that is something that we will be expanding on once we are, happy with the number of classifiers that we have. And like I said, we're really focused on the bright field right now. But yes, definitely nucleator is probably at the top of our list for things to do, just because we're already, finding the cell. So finding those edges should be pretty easy, hopefully.
Yeah, and if you remember, like we got like the mesh representation in like some of the views. There was like this kind of like mesh representation. So that that is yeah, like that's the software like kind of like showing you like some sort of like size. So yeah, as Elisa has mentioned, like definitely like the nucleator would be like kind of like the next obvious probe to implement in a Stereo Investigator AI.
Oh, and then so a good email to contact us is info@mbfbioscience.com. And then there's also going to be actually I'm just going to go to our next slide here. There will be a short survey after this webinar. So in that you can indicate where you're interested and include your email, and we will contact you as well. So getting in touch with us either way would work. So yes, if there isn't any other questions, we would like to thank you for joining us. If you think of other questions afterwards, please, you can visit our forum page to help get any answers, or reach out to us. Like I said, the info@mbf Bioscience would be a great email to use. And if you could please fill out the short survey after, that would be also very helpful and a great way for us to get in touch with you after this webinar as well. And also, if you want to learn more about what we have going on, you can be on the lookout for our office hours series to see what we have upcoming for this and other products. And also, if you have any requests, if you do have any of our other products, we would love to hear from you. We're all good, and we look forward to hearing from you and seeing your images.
Thank you so much.
All right, thank you,
Yeah.
everyone.
Bye bye.
Bye.